My name is Joseph Marks from Baylor College of Medicine. This talk is about stem cells. First, we'll examine what a cell is, then we'll examine what a stem cell is, then we'll examine the difference between embryonic and adult stem cells, and finally how stem cells could potentially be used to treat diseases. First, to understand what a stem cell is, we must first understand what a cell is. A cell is the fundamental, functional unit of life. For example, in humans, cells are the building blocks of tissues. Tissues comprise organs, and organs into body systems. There are over 200 different types of cells in the human body, each with a specialized function. However, there are some common characteristics between cells. Each eukaryotic cell has three principal parts: the cytoplasm, or the body of the cell, the nucleus, or the control center of the cell, and the DNA, which is the genetic material or the instruction booklet of the cell. The DNA contains all of the information necessary to build an entire organism. It also tells the cell what to do, how to do it, and when to do it. In addition, the DNA has all the information necessary to build an entire organism. We'll explore this point a little bit more in a minute. Here are two examples of what cells look like. On the left-hand side is a muscle cell, and on the right-hand side is a kidney cell. As you can see, they have distinctly different shapes or morphologies. These cells combine with other similar cells to make organs, and these organs carry out a specific function in the human body. Let us first review how a complex organism like a human determines how a cell becomes a specific type of cell. Remember that a sperm and egg join together, in which the sperm injects its nucleus into the egg. The two nuclei fuse together, and the cell begins to divide, a fertilized egg. From this single cell, an entire organism is created. So the question remains is, how does this kidney cell know to become a kidney cell? We can think of DNA as an instruction booklet. And though all cells can read certain chapters, a specific cell, like a kidney cell, can only read the chapter on kidney cell function. It cannot, for example, read the chapter on muscle cell function. More interestingly, cells can no longer read other chapters that do not relate to their function. So as I said before, a kidney cell reads the kidney cell chapter. A muscle cell cannot read the kidney cell chapter. And this is how cells differentiate or specialize into a very specific cell type to carry out a very specific job in the tissue. Now that we understand what a cell is, and we understand how cells specialize by selectively reading certain parts of their DNA, we can now examine what a stem cell is. In order to understand stem cells, we must first understand three very important terms regarding stem cells. The first is totipotent. Totipotent refers to a cell which can become any other type of cell in the human body, as opposed to pluripotent, which is a cell that can become many other different cell types, but not all the different cell types. Third is differentiation, which defines the way that a cell specializes into a very specific cell type. For example, how a cell differentiates into a kidney cell. It specializes to become a kidney cell. And the last term is plasticity, which means that a cell from one tissue can become the cell of another tissue. We can think of these terms as being more restrictive. A totipotent is the least restrictive, pluripotent is a little bit more restrictive, followed by plasticity, and then differentiation is the most restrictive term. So now that we understand the terms used to describe stem cells, we can understand what a stem cell is. A stem cell has two important characteristics. First, it's an unspecialized cell. In other words, it's a pluripotent cell that can become any other type of cell in the human body. Second important characteristic is that a stem cell must be able to self-renew. It must be able to generate more stem cells. Scientists refer to two distinctly different types of stem cells: embryonic stem cells and adult stem cells. As these different types of stem cells come from uniquely different sources. Where do we get stem cells? Let us first examine the origin of embryonic stem cells. Let's think back to fertilization, where we have sperm and egg fusing together and the cell begins to divide. Following the fusion of the two nuclei, the ball of cells, which is called the blastocyst, begins to divide. 2, 4, 8, 16 cells, until it reaches a ball of cells, which is called a blastocyst. This is still not implanted in the uterus yet, and it's a ball of cells totaling about 150 cells. And it is about 5 to 6 days old. The outer wall of the hollow sphere will become the placenta, while the inner cell wall will eventually develop into the organism. After the blastocyst forms, the cells start to divide into specific tissues, and so there's a point at which the inner cell mass then starts to differentiate into specific cell types. Embryonic stem cells are the pluripotent cells that are harvested from the inner cell mass that are still able to read all of their DNA. So they're harvested before the cells begin to specialize. In the laboratory, scientists harvest the blastocysts, remove the inner cell mass, and plate them on a petri dish, in which these stem cells can then be maintained in culture. These stem cells, as described before, are pluripotent. They can become almost any other type of cell in the human body. So what is the difference between embryonic stem cells and adult stem cells? First, adult stem cells come from different places. As indicated, adult stem cells come from adult tissues. There are many tissues in the body which have been shown to harbor adult stem cells, from bone marrow to the heart to the brain. Generally, it is thought that these stem cells are used to replenish dying cells in the human body through the normal wear and tear of things. In addition, adult stem cells are also thought to aid the body to help it repair after disease or injury. Though adult stem cells are very similar to embryonic stem cells, they are not as pluripotent as embryonic stem cells. So that adult stem cells may not be able to differentiate into all of the different cell types. But this is an area of intense investigation, and it is still undecided on whether or not adult stem cells can differentiate into as many cell types as embryonic stem cells. We know what a stem cell is. We know where we can get embryonic stem cells. And so what can we do with it? Once stem cells are maintained in the laboratory, scientists can coax them to become any other different type of cell in the human body. For example, they can coax them to become a blood cell, a muscle cell, or even a nerve cell. For example, a glial cell. These cells can then be injected into a patient, for example, with a spinal cord injury, and help possibly restore part or possibly all of the function. This has actually been done in mice, and researchers are currently trying to do this experiment in human trials. So now that we know how stem cells can be used to possibly be used in the clinical setting, who can benefit from stem cell research? There are more than 100 million Americans that suffer from diseases such as spinal cord injuries, heart disease, and diabetes, as well as cancer and a host of other diseases. In addition to the direct treatment of these diseases with stem cell research, stem cells can be also used for drug discovery and also to understand how a disease works, as well as basic cellular functions. One example of this is using stem cells to help screen drugs. Normally a drug is first tested in the laboratory, then it is tested in animals, followed by testing in humans. However, how a mouse, for example, uses a drug and how a human uses a drug is distinctly different sometimes. And so this poses a risk during human clinical trials. However, embryonic stem cells can be made into liver cells. These liver cells can then be used to test new drugs to first screen if these drugs are toxic to human cells. This will thereby increase drug development safety. Lastly, embryonic stem cells will help scientists learn about the fundamental workings of the cell, not only to further stem cell therapies, but also understand the basic fundamental disease mechanisms. In conclusion, we have learned that what a cell is, that a cell is the fundamental functional unit of the body. Stem cells have two principal characteristics, that they can become any other different type of cell in the human body, and that they have to be able to self-renew. There are two types of stem cells, adult and embryonic stem cells. Embryonic stem cells come from the inner cell mass of the blastocysts, while adult stem cells come from different adult tissues such as the brain or skeletal, or brain or muscle. Adult stem cells are not as pluripotent as embryonic stem cells, though this point is heavily debated. Lastly, stem cells can be coaxed to become any other type of cell in the human body, like muscle or brain cells, which can be used to treat a wide range of diseases, providing a new and exciting area of medicine.